Hello and welcome. The purpose of this module is to provide an overview of how to use a cloud-based modular system to analyze RADseq data for evolutionary applications. This module has been developed by the University of Wyoming INBRAE program. In this overview, we'll explore key concepts commonly used in RADseq or restriction site-associated DNA data analyses to help you better understand the analytic workflow. All the scripts used here are presented as a Jupyter notebook. And while we're introducing this as a cloud-based module system, it is important to note that the architecture is not specific to the cloud. So with only minor adjustments, you can run these analyses anywhere that support Jupyter notebooks. The module system is designed to simulate the workflow of a scientist starting with raw data. In this case, the raw data is our raw FASTQ sequence data, and running preliminary analyses in IPyRAD. And these foundational steps set up the data for a range of downstream evolutionary analyses, such as our population structure analyses and our phylogenetic analyses. Before we dive deeper, let's start with the basics of RADseq and the two main types of data sets you're likely to encounter when using this approach. The two approaches are basic RADseq or just RADseq and our double digest RADseq. RADseq, or restriction site associated DNA sequencing, is a technique used to sample and sequence homologous regions of the genome across many individuals. This process involves cutting DNA with restriction enzymes. The restriction enzyme cut sites are designated as X's here. So this process involves cutting the DNA with a restriction enzyme, attaching sequence adapters, and then sequencing those fragments located near those cut sites. Overall, it's a powerful method for discovering and genotyping genetic variants, especially in non-model organisms. A common variation of this method is called DDRAD, or double digest RADseq. As the name suggests, it uses two restriction enzymes instead of one. This dual digestion provides much more control over which genomic fragments are retained, making the results more reproducible and enabling precise size selection. So the main difference, traditional RADseq typically uses a single enzyme and relies on mechanical shearing to produce fragments. In contrast, DDRAD uses two enzymes and size selection to consistently target the same genomic regions across samples. This leads to better consistency and reduced genome complexity. RADseq has also important applications in biomedical research, especially in cases where traditional whole genome sequencing may not be practical or necessary. While it is used in biomedical contexts, it's somewhat limited compared to the other sequencing methods. It remains highly relevant in specific scenarios. For example, when whole genome sequencing is too expensive, especially for studies requiring large sample sizes, when working with non-model organisms such as wild populations of disease vectors or hosts, or when the focus is on discovering SNPs or understanding population structure rather than obtaining the complete genome assemblies. These types of studies can be incredibly valuable in understanding genetic diversity, identifying potential disease-resistant genes, and informing conservation or public health strategies. For this module, we'll be working with double digest RADseq data collected from the red diamond rattlesnakes. Our goal is to use this data to explore population structure across the Baja California region. And this case study provides a great real-world example of how to use DD-RAD data, or of how DD-RAD data can be applied to investigate genetic variation and population differentiation in a non-model organism with ecological relevance. For this module, we'll be running our analyses on the Google Cloud platform using a tool called Vertex AI Workbench. Vertex AI Workbench is a cloud-based environment that integrates seamlessly with Jupyter Notebooks. It's designed for data science and machine learning workflows, but it is also well-suited for genomic and bioinformatic pipelines like RADseq. The advantage of using Vertex AI is that it gives you a scalable computing resources, integrated data storage, and collaborative interface all within the cloud. That means that you can run your analyses efficiently without needing a high-powered local machine. However, you could run all of these analyses on a machine if needed. However, there are a few specific things to know when running this modular system within this environment. And we're going to walk through some of those in the next steps of this module. The first thing that you'll need to know is where to find the Vertex AI system within the Google Cloud platform. So if you click the three little dots on the left-hand side, it'll bring you up a menu that you will see Vertex AI. And then if you go within that, you will see the workbench. When you click on that, you will need to be able to, or you'll need to create an instance to be able to run all of these analyses. And so we would like to point you to the documentation linked within the README. But the one thing that is very important is that you use the correct container. When you create the instance, you will have to go to the advanced settings, go within the environment, and then click use a custom container and copy the correct Docker container image within that space. Other things to note are starting and stopping the instance. And so when you are looking at the instance that you've just created, by clicking on the details, you should be able to see a start and a stop button. This is how you start the instance of your machine. When it is start and stopped, it will tell you active for it being started and stopped for it being stopped. When the instance is active, you may open the Jupyter Notebook. When you open the Jupyter Notebook, this is what it should look like. It is important to note that I've already run through this modular system on RADseq Cloud Learning. And so I have a lot of things listed in my folder. You will need to go to the terminal to be able to copy this RADseq Cloud Learn file within your instance. So you can see that you can access the terminal there and then additionally follow the instructions that we've linked in the README on the GitHub. One additional thing that I wanted to note is to remember to stop your instance after you're done performing the analyses so you don't get charged additional fees for something that you're not using. And then finally, to cover the key concepts that we're going to be learning in the additional submodule videos, is learning how to process this RADseq data using IPyRAD. We'll go into a little bit more on IPyRAD and how that specifically works. We will also go into population or the first steps in analyzing population structure data. This is contained within submodules 1 and 2. And then within submodules 3 and 4, we'll also go over some of the key phylogenetic methods that we use, and how we plot them in R.